Hello and welcome everyone. In the immunology lecture series, today we shall continue with yet another interesting topic, that is the antibody structure and functions. So, let's proceed. The specific learning objectives of today's lecture is why antibodies are called immunoglobulins, what are antibodies, describe the structure of immunoglobulins. Identify the characteristics and functions of immunoglobulins. So let us first try to address the question why antibodies are called immunoglobulins. As you can see here, this is a serum electrophoresis run over here. So when the serum undergoes electrophoresis, there are 5 bands separated, and these 5 bands of proteins, all these 5 bands include proteins. They include the albumin followed by globulins. There are 4 types of globulins as you can see in this electrophotogram, that is the alpha 1 globulin, beta 2 globulin, alpha 2 globulin, beta globulin and gamma globulins. So the antibodies, which are also known as immunoglobulins, they are basically globulin proteins which are present in this gamma region, and therefore the antibodies are also known as immunoglobulins. Immuno stands for immunity, whereas globulin is a type of protein which is present in the plasma. Now we will try to define an antibody. So antibodies are a group of proteins collectively called immunoglobulins. These are glycoproteins in nature that react specifically to an epitope of an antigen. So it's very important for you to understand that an epitope is that region of an antigen which elicits immune response and which binds to the antibody. These antibodies constitute of 20% of plasma proteins. Now we will see the effector functions of antibody. So antibodies are present on the B cell membrane and are secreted by the plasma cells. So it's important for you to remember once again over here that it is the plasma cells or the B lymphocytes which secrete the antibody, and the type of B lymphocyte is known as plasma cells. The function of the antibody is to eliminate or neutralize the antigens by 3 processes chiefly, that is the phagocytosis, opsonization and antibody dependent cell mediated cytotoxicity. Now that you have understood what is an antibody, what is an immunoglobulin, let us take a look at the structure. Antibodies are glycoproteins in nature and they consist of 4 polypeptide chains that you can see here. There are 2 identical heavy chains and 2 identical light chains. The heavy chains are they have more number of amino acids and they are longer. Their molecular weight is around 55,000 Daltons each. The light chains are shorter with fewer amino acids and their molecular weight is around 22,000 Daltons each. The antigen combining site is present at the amino terminus. So being polypeptide chains, they have an amino terminus and a carboxy terminus. As you can see here, the antigen binds to the amino terminus region, and the carboxy terminus region elicits biological activities such as complement activation and opsonization. The light chains and the heavy chains are made up of variable regions uh, each and constant regions. The C, L and CH stands for constant regions of the heavy and light chain, whereas VL and VH stand for variable regions of the heavy and light chain. The four polypeptide molecules are connected to each other with the help of disulfide bridges. As you can see here, there are two types of disulfide bridges, the intermolecular disulfide bridge and intramolecular disulfide bridges. Now, the heavy chains are structurally and antigenically distinct from each other, and they are designated by the Greek letter corresponding to the immunoglobulin class. There are five types of heavy chains, and therefore there are five classes of immunoglobulins. Heavy chain gamma is for immunoglobulin G, alpha heavy chain for immunoglobulin A, mu heavy chain immunoglobulin M, epsilon heavy chain is in immunoglobulin E, and delta heavy chains are found in immunoglobulin D. The light chains are of two types, kappa and lambda. They can be only one type of light chain in one immunoglobulin molecule, and the most frequently found light chain is of kappa type. 60% of immunoglobulins have kappa type of light chain, and 40% immunoglobulins have the lambda type of light chains. So this was about the structure of antibody. This is a typical structure of immunoglobulin G. We will continue further. Now we will take a look at the variable and constant regions of an immunoglobulin. So the variable region you have been hearing since long. You must be wondering why it is known as the variable region. So the first 108 to 110 amino acids lying in the variable region of both heavy and light chain are highly variable. So the amino acids present in these regions are highly variable, and the amino acids present in the constant region are comparatively less variable. The number of amino acids present in the light chain are approximately 200, whereas the number of amino acids, total number of them present in heavy chain are 440. You know that the variable region has amino terminal, whereas the constant region has the carboxy terminus. Now if we take a deeper look into the variable region, we will see two more regions, that is the hypervariable and less variable regions. The hypervariable regions are the domains present on heavy as well as the light chains in an immunoglobulin, and they are directly in contact with the antigen. So the binding property of an antibody are determined by these hypervariable regions. There are 6 loops, and these, as you can see, that the heavy chain has loop 1, 2 and 3, whereas the light chain has loop 1, 2 and 3. So these are the um, uh, hypervariable regions, which are also known as the complementarity determining regions. Now what is an epitope and paratope? So epitope is that region of antigen which is responsible for its antigenic properties, and it elicits immunological response. It goes and binds to the antibody. And then comes the next question, what is a paratope? Paratope is that region of antibody which recognizes and binds to the antigen, especially with the epitope region. There is one more region called the Hinge region found in the immunoglobulin. The length of this Hinge region is variable. we will take a look at the hinge region and understand its function. So this is the immunoglobulin, and you can see that this antigen binding site fragment of the immunoglobulin is connected to the Fc arm with the help of hinge region, which are further connected by disulfide bridges. What is an Fab arm and Fc arm we will discuss in the forthcoming slides. The hinge region provides three-dimensional flexibility to the immunoglobulin so that it can bind tightly to the antigen. I will now quickly tell you about the structural components of immunoglobulins. As immunoglobulins are highly susceptible to enzymes, the structural components of an immunoglobulin were separated by enzymatic digestion. Here we will see action of two enzymes. One is the papain and second one is pepsin. When papain acts on an immunoglobulin, we receive three fragments. Two fragments of Fab region, which are known as the fragment antigen binding region, and the second region that we receive is the Fc region, which is the fragment crystallizable region. The fragment crystallizable region is insoluble in cold and it crystallizes, whereas the Fab region is a soluble fragment and it is unable to precipitate with egg albumin. Each molecule of immunoglobulin is split by pepsin and papain, and the fragments received are of two different types. For pepsin digestion, we get two Fab fragments which are joined together, and we get small pieces of Fc fragment. The Fc portion can be digested by pepsin into various small fragments. Now we come to immunoglobulin classes. So based on the physicochemical and antigenic differences, the five classes of immunoglobulins have been recognized, which are namely immunoglobulin G, A, M, E and D. So there is a mnemonic for you to remember this, and that is gamed or gamde. You can use any one of the mnemonic. Now we will take a look at the valency of immunoglobulins. So there are different valencies of each immunoglobulin. Valency means the ability to bind to number of antigens. So if there is a single immunoglobulin, that is monomeric immunoglobulin, which includes G, E and D immunoglobulins, they can bind to two antigens as they have two antigenic binding sites. The dimeric immunoglobulin is immunoglobulin A. It is joined by the joining chain or the J chain, and it has two units of immunoglobulin, so the valency will be 4, 1, 2, 3 and 4. It is complex with a protective secretory component, and it is found in all secretions including the colostrum. The pentameric immunoglobulin is the immunoglobulin M, which have 5 units of immunoglobulins bound together by the J chain, and immunoglobulin M monomers linked by disulfide bridges and one J chain. So they can bind to 10 antigens, and therefore the valency of immunoglobulin M is 10. Valency of immunoglobulin A is 4 and valency of immunoglobulin G, E and D is 2. Now we will discuss the major functions of immunoglobulins. So we'll start with immunoglobulin G. Here we will follow the mnemonic GAMDE, Gamde. So immunoglobulin G major function is that it's a main antibody of secondary immune response. It opsonizes bacteria, making them easier to phagocytose. It fixes complements, which enhances bacterial killing. It neutralizes bacterial toxin and viruses, and it crosses the placenta. So it is the only immunoglobulin that can cross the placental barrier, and it provides immunity to the newborn. Immunoglobulin A is the secretory immunoglobulin A. It prevents attachment uh, of bacteria and virus to mucous membranes, and it does not fix complement. Immunoglobulin A is present in various secretions which include milk, mucus, etc. Immunoglobulin M is produced in primary response to an antigen. It fixes the complement. It does not cross the placental barrier as it is pentameric and it is very heavy. Uh, Antigen receptor on the surface of these cells. Immunoglobulin D is found on the surface of many B cells as well as in the serum. Immunoglobulin E mediates the hypersensitivity by causing release of mediators from mast cells and basophils upon exposure to any allergen. It defends against worm infections by causing release of enzymes from eosinophils. It does not fix the complement. The main host defends against helminth infection. So these were some of the major functions of different immunoglobulins. These are my references for the lecture. Thank you for watching. Like, comment and share and subscribe to my channel.